Hey everyone, and welcome to another deep dive. Today we're looking at a recent scientific breakthrough that's it's pretty amazing. Scientists have created the first complete blueprint of the human spliceosome, which is kind of a big deal.
It is a big deal. You know, most people have never even heard of the spliceosome, but this is like the most complex machine we've ever found in human biology.
Wow. Okay, so I have to admit, I don't really know much about the spliceosome either.
Well, it basically edits your genetic code.
Mhm.
So every gene in your body, almost every gene, relies on the spliceosome to work properly.
Okay, so it's like a molecular editor that makes
Yeah.
sure your genes are, functioning correctly.
Exactly. And when it makes mistakes, things can go wrong, like really wrong.
You mean like diseases and stuff?
Yeah, we're talking cancer, neurodegenerative diseases, genetic disorders.
Oh, wow. Okay, so this blueprint could be huge for treating those diseases. But before we get into that, can we back up a bit and just explain what is the spliceosome exactly?
Sure. So you know how DNA is like the master blueprint for building a human?
Right.
Well, that blueprint needs to be translated into instructions that your cells can actually use.
Okay.
And that's where RNA comes in. It's like a messenger, carrying genetic information from the DNA to the protein-making machinery in your cells.
So RNA is like the construction crew getting directions from the architect.
Exactly. But here's the thing. The initial RNA transcript is kind of messy. It's got these extra bits of information called introns that need to be removed before the final protein can be made.
Introns.
Yeah. They're like extra scenes that got left in the movie. They need to be edited out.
Oh, okay.
And that's where the Spliceosome comes in. It's like a team of Hollywood editors, cutting out the unnecessary scenes, the introns, and splicing together the good stuff, the exons, to create the final polished version of the RNA.
So the spliceosome is basically deciding which scenes make it into the final cut of the protein movie. That's pretty cool.
It is, and it's incredibly efficient. Humans have roughly 20,000 protein coding genes, but thanks to the spliceosome, we can produce over 100,000 different proteins from those genes.
Wait, 100,000 proteins? From just 20,000 genes?
Yep.
That's amazing. How is that even possible?
That's the magic of splicing. By rearranging and combining different exons, the spliceosome can create a huge diversity of proteins from a relatively small number of genes.
So it's like having a wardrobe with just a few basic pieces of clothing, but being able to create hundreds of different outfits by mixing and matching them.
Exactly.
That's a great analogy. So how did scientists manage to create this blueprint of such a complex machine?
Well, they used a really clever approach. They altered 305 genes related to the spliceosome in human cancer cells, one gene at a time.
305 genes?
Yeah. And then they observed how those changes affected the entire genome.
So they were basically poking and prodding the spliceosome to see how it reacted.
Exactly. And what they discovered was truly groundbreaking. Each component of the spliceosome has a very specific job. It's not just a random jumble of molecules bouncing around. There's a highly specialized division of labor happening inside this tiny machine. Yeah. Each protein plays a unique and essential role, ensuring the entire process runs smoothly.
So it's like a perfectly choreographed dance, with every molecule hitting its mark at the right time.
That's a great way to put it.
Can you give us an example of what some of these specialized roles are?
Sure. They found one component that acts like a security guard, preventing the spliceosome from acting too early and potentially messing up the RNA sequence.
So it's like making sure the editors don't start cutting the film before they've even watched it.
Right. And another component acts as a guide, helping the spliceosome identify which RNA segments need to be removed.
So it's not just about cutting, it's about knowing what to cut.
Exactly. And yet another component is responsible for making sure the cuts are made in exactly the right places, ensuring the final edited RNA is accurate and complete.
This is incredible, and it has huge implications for how we think about diseases, especially cancer.
Absolutely. And that's exactly what we'll dive into next.
Okay, so we've talked about how the spliceosome is this complex little machine that's responsible for editing our genes. But you mentioned that when it makes errors, it can lead to diseases.
Right. Errors in splicing are linked to a lot of different diseases, actually. Cancers, neurodegenerative diseases, even genetic disorders.
So it's like a film editor accidentally cutting out a crucial scene or splicing in the wrong footage. The final movie just isn't going to be the same.
Exactly. And in the case of our bodies, that could mean a protein doesn't function correctly, which can lead to disease.
And you mentioned cancer specifically.
Yeah, cancer cells are kind of notorious for their ability to adapt and grow uncontrollably, and they really rely on the flexibility of a spliceosome to do so.
So they're basically hijacking the splicing process to
Mm,
produce the proteins they need to survive?
exactly.
That's kind of scary.
It is, but it also presents a unique opportunity.
Oh, wow. So?
Well, because cancer cells rely so heavily on the spliceosome, targeting the spliceosome to be a really effective way to treat cancer. Remember that SF3B1 component we talked about earlier? The one that acts as a security guard, preventing the spliceosome from acting too early.
Yeah, the one that makes sure everything happens in the right order.
Well, it turns out that SF3B1 is frequently mutated in several types of cancer.
Oh, wow.
Yeah, melanoma, leukemia, breast cancer.
So these mutations are messing up the whole splicing process.
Exactly. And the researchers found that altering the expression of SF3B1 in cancer cells caused a whole cascade of failures throughout the splicing network.
So it's like a domino effect.
Right. One mistake leads to another.
Yeah.
And another, until the cell can't function properly anymore.
So by targeting these faulty components of the spliceosome, we could potentially develop new cancer treatments.
Exactly. And what's really interesting is that this approach could be even more effective than traditional DNA-focused treatments.
Oh, why is that?
Well, because cancer cells are already pushing their splicing machinery to the limit. They're constantly adapting and evolving, but that also makes them really vulnerable to disruptions in splicing.
So it's like,
Yeah.
if you try to fix a car that's already running on fumes, one more problem and the whole thing just falls apart.
Exactly.
That's a great analogy. So this blueprint could be a game changer for cancer treatment. But what about other diseases that are linked to splicing errors?
Well, the good news is that this blueprint gives us a really detailed roadmap of the spliceosomes, so we can pinpoint exactly where splicing errors are occurring in different diseases.
That's amazing.
It is. It's like having a troubleshooting guide for your genetic code.
Are there any examples of how this could lead to new treatments?
Sure. We've already seen some incredible successes with splicing-based treatments for rare diseases, like spinal muscular atrophy, which is a genetic disorder that affects muscle development and movement.
I've heard of that.
Well, there's a new drug that targets a specific splicing error, and it's helping a lot of children with this condition live longer, healthier lives.
That's incredible. So it sounds like we're on the verge of a whole new era of medicine, where we can target the root cause of diseases at the genetic level.
I think so. And this blueprint is a huge step in that direction.
What are the next steps in this research?
Well, the blueprint itself is a powerful tool for future research. And the best part is, it's publicly available.
Really?
Yeah. Scientists all over the world can use it to study the spliceosome in more detail, identify splicing errors in various diseases, and develop new therapies.
That's fantastic. It's a really great example of the collaborative nature of science.
Yeah.
So what's next for splicing-based treatments?
Well, I think we're going to see them move beyond rare diseases and into more common conditions like cancer, heart disease, neurodegenerative disorders.
The possibilities seem almost limitless.
They really do.
Yeah, it really is amazing to think. We've only just started to understand what this tiny machine can do.
Yeah, and it's exciting to think about what we might be able to do with this knowledge in the future.
Right. Like you said, it's a paradigm shift. We're not just treating symptoms anymore. We're actually trying to understand and fix the root causes of disease.
And that's really powerful.
And the implications go way beyond just treating diseases. I mean, if we can manipulate the spliceosome, could we prevent diseases altogether or even enhance human capabilities?
That's a really interesting question, and I think the answer is potentially yes. If you think of the spliceosomes as kind of like a team of editors shaping our genetic stories, this blueprint gives us the power to actually rewrite those stories.
Wow. That's incredible. We could potentially correct genetic errors before they even cause problems or enhance certain traits.
Yeah, the possibilities are pretty much endless.
It's both exciting and a little bit scary.
I agree. It's important to consider the ethical implications of this technology, but it's also important to celebrate the incredible potential it holds.
Yeah, absolutely. So to recap, we've been talking about the spliceosome, this amazing little machine that's responsible for editing our genetic code.
And we've seen how its precise workings are crucial for our health, and how even the tiniest errors in splicing can lead to serious diseases.
But we've also learned that scientists have created a blueprint of the spliceosome, which could revolutionize the way we treat and prevent those diseases.
This blueprint is a testament to human ingenuity and our desire to understand the world around us. And it's a reminder that even the smallest things can have a huge impact on our lives.
I love that. So for our listeners who want to learn more, we've included links to the original research and some additional resources in the show notes.
Be sure to check them out.
Thanks for joining us on this deep dive into the spliceosome. Until next time, stay curious.